So welcome everybody and thank you for coming. As we begin, please note that this lecture is being recorded. I am John Lors, Director of the National Institute of General Medical Sciences, and I want to welcome you to the DeWitt Stetton Jr. Lecture. The lecture itself honors DeWitt Stetton Jr., who was the third director of NIGMS and had a strong commitment to basic science, especially research in genetics, cellular, molecular biology, and chemistry. He spent most of his career at NIH serving in 2 institutes and in the office of the director of NIH, and was particularly well known as a co-author of the early editions of the textbook Principles of Biochemistry. In recognition of his many achievements and contributions, both within and outside of NIH, he has the rare distinction of having 2 NIH entities named in his honor. This lecture, as well as the DeWitt Stetton Jr. Museum of Medical Research, which I actually just learned today is not a physical museum, but is all of the exhibits about science that you see scattered around the campus, including the ones that are right outside of this lecture hall. The way the lecture works is that the speaker, Dr. Rai Young, will give a 50-minute talk on his research, which will be followed by a 10-minute question and answer session. You can ask your questions by using the mic in the room if you're here with us in person, or by selecting the Send Live Feedback button on your videocast screen if you are joining us virtually. If you are seeking a continuing medical education credit or CME, the code is 50098. Let me repeat, 50098. So today's speaker, Rai Young, joins us with a very distinguished list of Stetton lecturers. He is a founding director of the Center for Phage Technology and a distinguished professor emeritus in the Department of Biochemistry and Biophysics at Texas A&M University. Rai has dedicated his career to understanding bacteriophages, especially how they lyse bacterial cells. In fact, much of the modern field of phage lysis comes directly from his research. Even when others turned away from the field, he saw the potential to use phages with their facile genetics, molecular biology, and biochemistry to ask specific and important biological questions. During today's lecture, Rai will discuss how phages control lysis in a time-dependent fashion and how they might be harnessed in developing new antibiotic therapies. Rai received his undergraduate degree in biochemistry from Rice University and a PhD in molecular biology from the University of Texas, Dallas. He's also a veteran of the Navy, and I think he probably is the Stetton lecturer and maybe the Walls lecturer who has the most impressive journey for how he actually got here today. He didn't fly, he didn't take a train, he actually, with his wife, drove or piloted his boat all the way from Texas through the Gulf, through Florida, up the waterways, and into the Chesapeake Bay, where
10 miles per hour is one.
So that is some serious dedication to get here. So thanks for coming, Rai. It's great to have you with us.
Thank you very much. So, first of all, I'm sorry about wearing my glasses like this. I'm not trying to imitate the old fuddy-duddy professor, but I lost my bifocals, and I have to be able to look down. My wife says I look ridiculous. I hope they don't fall off. So this slide is just to make sure that I acknowledge the people who've done so much of the work here. Several of them are here in the building, and I'll come back to it at the very end because this is kind of the work product. This NIGMS grants funded this work for over 40 years, and part of the product is well-trained people who've gone on to success in science in many ways. The ones in red are all faculty members or a founder of a biotech company, and almost all the other ones are on their way there. So this, to me, phage biology has always been a great system for training because you can get experiments done so quickly. So I think that's why, even though nobody there is still working on phage lysis or even phage, it's been a good system for that regard. Okay, so it's a great honor to be over 40 years ago before this started. There's still not been, as far as I can tell, I'm the first phage guy, which is really weird since NIGMS is the birthplace of funded phage biology. The only other person who might have given a phage talk was Mark Potashny, but I think he was talking about yeast transcription at that time. So we're writing a historical wrong here. All right, so I can remember, this is a reconstruction, not the actual, but back in 1969 or 70, I was, no, it was even earlier than that. It was later than that. It was 2, I think. I was standing in John Beckwith's lab at Harvard Medical School. No, I'm 5 years off, 77. Old fuddy-duddies there have trouble with decades. At any rate, I was standing there. I had borrowed his shaker flask and his spectrometer to follow some lambda inductions. Now, I won't tell you why I was doing these lambda inductions. With lambda, you can heat shock the bacteria that are growing in a culture flask, and then the prophage will suddenly induce, and then you get synchronous infections this way, and that's what the real value is. So I had started 5 cultures and induced them, I thought, at all the same time. I looked down and discovered 3 minutes ago that I had not started one of the cultures. So I shifted it. So one of these 2 flasks started 3 minutes before the other. Otherwise, they're identical. So if you look at it, all the cells in the original flask have lysed the phage, and none of the cells in the original flask have lysed in the late one. So this tells you that phage lysis, at least as enacted by lambda, is very tightly controlled in terms of time. So this is an event that can keep time on the order of low minutes to seconds, and I've had children that can't keep track of time in days. So this turns out all to be due to a single protein of 105 amino acids, and that protein is a holin. And almost all of this lecture, and by the way, this is a lecture, so you're going to hear mostly my theories about what's happening with some data but no controls, and then I will try to cover the entire how my understanding is of the entire system that makes going from there to there. And this is, let's see if I can not crash the computer. This is what that looks like at the single cell level. And what you should see, this is all in seconds here, so we're looking at a millisecond view of what's going on. Notice that it's not just a general blowout of the whole thing. There's a specific lesion that occurs at one point in the cell. We call this local blowout. And so that whatever that timing event that we looked at the macroscopic level occurs also at the single cell level. This happens at a single place at a single time. And for several seconds after that, the rest of the structure of the cell is normal. And that blowout, presumably, although we can't see it, all the 100 or so phage particles are expelled from that end of the cell in this case. About 80% of the time, it does occur at the pole. This was done by Jesse Cahill, who's now got his own lab at Sandia. So I included this lab, this data, because I'm pretty certain that Shankar was there when they did this. So this is actually not lambda, this is T4. So it's unusual for me to show T4 slides. I'm a lambda tribe person. But the T4 Delbruck group did this experiment. Basically, you infect T4 at time 0 into E. coli, at an MOI where every cell is infected. And notice that very sharp temporal control of lysis exists. It's at a different time because this is a different phage, but it's very abrupt. And it's the graphical representation of what we already saw. So one other thing we have to explain is the universal character of phage-induced lysis. And that is, if you add cyanide early in infection, it just stops all energy metabolism and nothing happens. If you look inside the cell, phages aren't replicating, there's no assembly going on, nothing's happening. And that shows up as no change in OD for a long period of time. But if you wait, say 15 or 20 minutes, and now inside the cell, phage particles are being assembled, there's robust phage gene expression, the lysis proteins are all being expressed. The instant you add cyanide or any uncoupler like dinitrophenol or anything else that can depolarize the membrane, you trigger lysis immediately. And this is called premature lysis. That terminology was invented by Max Delbruck. And it's a universal phenomenon. So anytime when we're trying to figure out what holins do and how they do it, we have to explain not only how the timing is kept accurate within minutes or so. In addition, you can disrupt that timing prematurely by collapsing the membrane potential. That's all you know. I'm sorry I'm so hoarse. I've answered so many questions today. Okay, so the model has to explain both things. All right, so this is the lambda lysis cassette. So if you drawn to scale, this is at the right end of lambda. Working, yeah. So there's the late promoter, and the first genes are the lysis genes, a little counterintuitive, and then there's the head genes and the tail genes, and this would be all the way into the next building. for the rest of the 20 or so odd genes needed. But all from one transcript. It turns on at about 8 minutes after infection, and it makes it very easy to study in terms of the kinetics, because it all is not, there's not a complex transcriptional. So this is either on or off, and after that it's just a matter of copy number. And there are 4 genes, S, R, RZ, and RZ1. We're going to spend most of our time talking about the genes are arranged, by the way, in the order of assaulting the envelope. So the first gene, gene S, get this right. Gene S actually encodes 2 proteins. It's 107 codons long. It makes one protein the full length, and that's the actual anti-holin. It also makes a shorter protein by 2 codons earlier initiations, and that's the holin, and that'll be the star of this show. We're not going to be talking about how S107 works, but you can tell from its name that it works by directly inhibiting the function of the holin. We'll briefly talk about that at the very end of the seminar. The next gene, and it targets the inner membrane. The next gene is the endolysin, or the lambda lysozyme. It's the enzyme that degrades the cell wall, and of course it attacks the peptidoglycan. And then weirdest genes out here, there are 2 genes in the same DNA, RZ and RZ1. They're 153 and 160 codons long. They're completely different proteins. They're both required, which raises all kinds of evolutionary quandaries. And they're encoding the 2 subunits of the spannin protein, and they're required for disrupting the outer membrane, which is also essential for lysis. So important to note that you can either use the phage, or you can clone those 4 genes into an inducible vector, and you get the same phenotypes. You change the absolute timing a little bit just based on copy number, but everything else, it doesn't matter if there's no other genes in the phage that have any effect on its function or timing. So sometimes I'll show you data from phage, and other times from plasmids, and I probably won't even tell you when I'm doing that. Okay, the first thing to do is to get rid of lambda lysozyme as a protein of interest. So this is the basic structure of the bacterial cell wall. And all you need to know is among the 4 most important scientific tribes, the T7, T4, lambda, and T5 tribes, each one of them had a different gene called lysozyme. And so it turns out that all 4 of these lysozymes are completely different enzymes. So lambda makes a transglycosylase, and T4 makes a glycosidase, which hydrolyzes the glycosidic bond. T7 makes an amidase that cleaves the bond between the peptide and the cell wall and the glycoside. And then T5 makes an endopeptidase. So they're all different enzymes. They're not related to each other. And they're all called lysozymes. And they're not much interest. In fact, they're interchangeable. They're always produced in, lysozyme is produced in 100-fold excess. And they're interchangeable, and they're boring. So the interesting guys here are the holins. So I made the word up, holin, so I'm only allowed 1 word, I think. If everybody generates a word, then that's too much additions to the English language. But so lambda is, this is to emphasize the fact that all holins share at least one thing. They all have at least 1 transmembrane domain, which is not surprising if it's doing something to the inner membrane. And then, so lambda, it has 3 transmembrane domains. Its amino terminus is out, and the C terminus is in, and this is the topology. The other 4 topologies, these are the only 4 topologies that have been experimentally confirmed. So there's type 1, 2, 3, and 4. If we'd been smart, this would be type 1, that would be type 3, because then the type number would be the number of transmembrane domains, but we didn't do it smartly. So type 1, I have 3, and there are, last time I looked, which was last week, there are 217,000 records in the identical protein group. And I checked, my expertise is not in biophysics, but I looked at the first 100 or so, and they're all different. So there are thousands, at least thousands of unrelated gene families, and they all have at least 1 transmembrane domain. But notice, too, that one of them, which is the T4 holin, which is also shared by T5, has a periplasmic domain of about 100 amino acids. And this is a signal transduction receptor. And this is the only case we know in biology where external information is directly input into lysis timing. Unfortunately, we're not going to be able to talk about this unless I get a pregnant question near the end. But it's a wonderful story that's recently made progress at the structural level. But it's not related to the basic story of how the timing occurs. So we remember that this is only one of the two proteins made by the lambda S gene. It's S105, which is the true holin. Okay, so the initial, back when I was starting to get my lab together, I got at least on a couple of papers and grant reviews that the simplest idea was that the holin, it was known that cyanide could cause phage infections. You had cyanide, you collapse the membrane potential, everything lyses. So that was already known. It was 1951. So the idea was holins just accumulate in the membrane, titrate out the membrane potential like it's well known. If you overexpress membrane proteins, you poison the membrane. And then when the energy was zero, that would be the same moral equivalent of adding cyanide. And so you get whatever was happening the same in normal induction as in premature lysis. So we answered that question quite a long time ago. This is work done by Angelica Grundling, who's now at Imperial College London, and a collaborator with Mike Manson at Texas A&M. So see if I can do this. So imagine these are cells that are tethered to the membrane by their flagella, tethered to the glass slide by the flagella. And so you're getting a direct readout of the membrane potential because the flagella, by the way, these flagella are locked in a clockwise orientation. And so we know their PMF is 170 millivolts right now because they're spinning at a normal rate. And now within a few seconds of each other, they both stop. And then in another 30 or 40 seconds, they're going to lyse. And it turns out that 25 minutes before we started this real-time video, we had added an inducer to turn on the lambda lysis genes. So what we saw was another representation of that same timing event before, where after a long period of time, in the order of minutes, 25 minutes in this case, this was a plasmid, the holins had been accumulating, and suddenly, instead of titrating out the energy potential slowly, it all happened at once. So we call this paper holins kill without warning. So it's a direct refutation of these kind of puke out model that everybody was suggesting. Okay, so I'm a geneticist, and lambda S genetics are really fun. Lambda genetics are fun, period. But lambda S genetics are even more fun. Now, this was actually an accidental experiment done by Angelica. She was trying to put cysteine, there's a single cysteine in S, and so by eliminating that, you can put cysteines anywhere you want. And the idea was to use these cysteines for various types of biochemical reactions, including accessibility studies, dimerization, oxidation, and so on. So she went through and put single cis residues. These are all missense changes. And we noticed, so this is color-coded mutations in the first, second, and third transmembrane domain, that every single missense change caused a timing change in lysis, every single one. Sometimes they were earlier, sometimes they were later. And we call this the rainbow lysis slide because this really got us thinking about how this timing clock could work. So that means that, and then superimposed on this, if you actually, this was done by inducing a lambda phage in trans to this plasmid. So if you actually counted the particles that were being generated, and remember, the particles are being assembled all the time that this is going on. Normally, at about 50 minutes, you'd have 100 or so. But if you go to 75 minutes, you have 200. If you go out here to 300 minutes, 3 hours, you'd have 600. So it does raise the question, why does lambda quit at 100? Is 100 a nice round number? It says that's good enough. Or what's the rationale for it? And the other thing is, how can we explain every single change in three different transmembrane domains grossly altering the lysis timing? And by the way, we checked and none of them have any effect on accumulation of the protein. So that led to a project that Rebecca White ran. And Rebecca she's now the CEO of a company called Avalo. I think it's the third one she's run. But she made this into a standard undergraduate lab. So every undergraduate who came in the lab would get a missense mutation. And so we had two original mutations from our original genetic screen back in the 70s. And these were alanine 52 to valine and alanine 52 to glycine. So these are very, I guess, they're conservative in one sense, but one's getting to be a large alkyl group and the other's a hydrogen atom. And neither one of them, so both of these mutations were isolated as non-plaque formers, right? So it turns, that's a formal proof that the holin has two functions, because you can knock out both of them, and either one of them is required. One is to cause the lysis, and the other is to cause the lysis at the right time. So if you look at this, so what she first started out was to have every single possible other amino acid change done at alanine 52. And what we discovered was that a number of them, 4 or 5 of these alleles, lysed earlier. Phenylalanine lysed at a ridiculously early time. No phages are produced by any of these, and that's why you don't get any plaques. But you also got a number that were slower, and if you put charged residues in the middle of the membrane, you're going to kill the membrane. So that wasn't surprising that a lot of the proteins were inactivated. And so then she extended that to the three residues next to that, 49, 51, 49, 50, 51, and 52. And basically the bottom line is the same for threonine 49 and 52, but in Met 50 was relatively silent. The only most changes either inactivated the protein or delayed it, but we got very large number of early lysis mutants at position 49 and 52. So the take-home lesson is that lambda can easily explore lysis time space. If it has a reason to explore lysis time space, the lambda holin can do that by single misses changes. And so a single conservative change can dramatically alter timing either earlier or later. Okay, so here comes the first model. So the model for how holins work is that the holin accumulates in the inner membranes. I'm not showing you anything about the phage. The phage particles are being, as long as there's lysis proteins being produced, that's also the morphogenetic period, so you're also getting phage particles produced. But we're not showing that in this cartoon. So the blue is the holin, and the lysozyme is the green, and they just accumulate, and suddenly they reach what we call a critical concentration. This is the model anyway. And then at that critical concentration, the proteins aggregate into what we call a raft, and then somehow that raft is converted into holes, and I'll show you in a little while that these holes are, they're unfortunately called S-holes. Never fails to. Anyway, these holes are micron scale. They're the largest, as far as I know, the largest holes formed in biology by normal means. And so these giant holes allow the lysozyme to escape and then attacks the cell wall. And then, of course, we already saw that lysis is fundamentally a punctate event. So that punctate event is defined by where that hole is formed. Okay. So remember, the universal holin phenotype is that premature triggering also has to be explained. So what we argue is that if you add cyanide at this point, instead of going through the waiting for the critical concentration before you form the raft, the addition of cyanide instantly allows whatever the level of holin is to form the holes. And it turns out one side prediction of that is that the holes should be smaller because there's less lysis protein, and as you'll see, that's the case. All right, so the first thing we wanted to show was is true that holin is actually mobile. And so in work Rebecca White and work collaborated with people from Kit Fogliano's lab. These are fluorescence bleaching experiments. So if you turn on the holin GFP fusion, which works pretty well, and if you use the A52V mutant, it never does anything but just stays mobile. So when you irradiate it, it immediately becomes refluorescent. And if you do the bleaching in the S105 GOP before triggering, early in the system, you get repopulation of fluorescent spaces. But after triggering, it's no longer, it doesn't recover, as you can see. This thing is not working now. So S105 is mobile before triggering and immobile after triggering. This was probably the hardest experiment we ever did. This is S-biochemistry. I won't go into the desperation modes that you have to do to purify holins. Turns out we complain about lambda where you can get 0.5 milligram per liter, but most holins you can only make in low microgram quantities per liter. But we were able to purify the lambda holin, and I actually have to tell you, we had to explore 13 different positions to put his tag in before we found one that would work. So this is work done by John Deaton and Dave Smith. So you basically purify it, you dump it into a chaotrope where it's totally unfolded, then you dilute that down into a suspension of liposomes that have fluorescent calcine in them. And what you see is the wild type releases the fluorescence. The mutant doesn't, even at twice the concentration. And even more importantly, purified material from a TS allele, which is allo-23 to threonine, works at the high temperature, but not the low temperature, which formally excludes the possibility that there's some contaminant of our preparation that was causing the permeabilization. So S does what it's supposed to do. It can permeabilize membranes in an allele-specific manner. And this is, I think this is, so just to remind you what happens at the milliseconds level. There's going to be a punctate expulsion of the cytoplasmic material at the local blowout. Okay, so our next goal was to visualize the S hole, and it should be big enough because it's micron. Well, this is how we learned it was micron scale. This was a collaboration with Andreas Holzenberg's lab at TAMU, and Christos Savos and Jill Dewey from my lab did the work. And so basically, you're looking at interruptions in the membrane of the E. coli, and the average size of these. Actually, let me show you the next slide, which is So this is actually a tomogram, and it's artificially. It took us a year to do this experiment, and then a reviewer required another one. It took another year to do the experiment. We finally published it. But this was about a 100-micron hole. On average, these holes, there's 1 to 3 of them per cell, and on average, they are about 350 microns. So everybody wanted to see the hole, and we finally did it. Okay, so then the next thing was, what's the structure of the hole? And so we knew that the S protein started out as a mobile, probably dimeric form that floated around in the membrane. It ends up in an immobile, large micron scale hole. But how is that? What's the actual structure and what's facing what? So what we wanted to do was to find out in the S molecule. Remember I told you about Angelica's work putting cysteines all over the place. So this is called the cysteine accessibility studies. So you can see all the cysteines we use. And then you treat this with a reagent, and then it blocks. And if the reagent modifies the protein, that means it's in the soluble phase, and then you can detect that by Western blots because you pegylate afterwards. The bottom line was that there were half of TMD1 and TMD3 that were exposed in the hole, were exposed to the aqueous solution, whereas in the membrane, whereas TMD2, no position in TMD2 in the middle was exposed. So that allowed us, and Kenneth Toe is the one who now runs Divergene, was the one who did this work. So this led to this model where the hydrophilic faces of TMDs 1 and 3 in the dimer, and I'm not showing you the evidence it's a dimer. It was very simple cross-linking experiments. So based on what we just saw, it's most consistent with the notion that the hydrophilic faces in the mobile dimer are facing each other, sequestered from the membrane. So we're looking down on the membrane now, right? So this accounts for the ability of the protein to freely migrate because its hydrophilic faces aren't facing any other side. So in the lumen, in the final structure, after collapse, this is the model that we would say, because we've shown that half the face of 1 and 3 are accessible to the soluble reagent. Okay, and by the way, we've measured the amount of S protein by quantitative Western blot and also by simply measuring the perimeter of the holes. And if you build on the fact that each one of these alpha helices is about 1 nanometer in diameter, then there's a consistent number of about 5,000 S105 molecules in the total perimeter of the holes. Okay, so here's the grand centralized model, I guess, and that is you start off with this mobile dimer. Actually, we have a mutant that doesn't dimerize and is very unstable, so we suspect that, well, we don't really know what controls the dimerization, but so the dimer is the molecule that undergoes the timing, because it's not until the dimer reaches a critical concentration that it suddenly aggregates to form a raft. And we're arguing that the total population of S molecules in the rafts is about 5,000, and it's the existence of the raft that causes the next phase, which is, and here's where I'm going to beg your indulgence. So what we think is happening is you have you have a short circuit because you have a relatively large surface of lipid-depleted adjacent protein molecules, which are featured by transmembrane domains, each one with an active polar, several active polar units. So we think there's a short circuit local depolarization in the raft, and that raft, now you have local depolarization, and now you're back to our original idea. If you depolarize the membrane, that causes the secondary, tertiary, and quaternary change that starts the formation of the hole. So it's the formation of the raft that leads to the first small holes, which then rapidly expand in a feed-forward mechanism to the large-scale 300-micron holes. So that's the grand model. And then either premature lysis or a lysis mutant that had an earlier triggering, which we saw was easy to create, simply causes the critical concentration to be lower, and so you end up with a smaller hole with fewer S proteins on the perimeter. And we've published that. That's exactly what happens. The mutants are premature triggering. You reduce the size of the holes. Okay, so what do we mean by critical concentration? I did not make that word up. So this work done by from Mark Krebs's lab quite a long time ago. He was from Karanda's lab, and he worked on the purple membrane protein, bacterial rhodopsin. And it's easy to purify. It's made in massive quantities, right? And they discovered very early on that if you purified it and then diluted it into a detergent of some percentage, then it would form. Is it finally not working at all now? Whoops. So the bottom line is that at whatever concentration you made the protein, at low concentrations they would all be in monomer, bacterial rhodopsin monomer. But as you went to higher and higher concentrations, these are sucrose gradients. You get more and more of it in the lattice form. So if you plot the amount that's in the lattice versus the concentration, you get a curve like this. And where it intersects the line is the critical concentration. It turns out that, so they had a rough cryo-EM structure of how the purple membrane was interacting with the units were interacting with each other. And they did some saturated immunogenesis. And so normalized to the wild type, I've highlighted two that are simple, relatively conservative changes that either change the critical concentration 5-fold lower or 6-fold higher, which when it does that, it completely changes the percentage that completely changes the critical concentration. So I hope that validates at least a little bit in your mind that this idea that it'd be easy. It's the fundamental reason why single amino acid changes in the holin, you're simply changing the concentration in which it suddenly forms the rafts. Okay, so now the final act of this is to show that it actually behaves like we say it does. So these are SGFP experiments where the GFP is attached with the linker to the C-terminal end cytoplasmic domain of the holin. And these are just now it's just now coming out in MBio, so it's relatively new. Let's see, can I get this going? So the idea is that you will see the whole formation suddenly. First, we start with dispersed SGFP, and then you will suddenly see them aggregate. In this case, at both poles, into rafts, which then disappear, and we think the disappearance of the raft is when the elongation occurs, and then you get the explosive disruption of the cell at both these positions. Sooner or later. This is only seconds. It seems longer when I'm looking at it. There we go. It looks painful, actually. Okay, so here's another one. This is a little more convincing because it's at different places besides the hole. About 80% of the time it happens at the pole. So in this case, several of them formed and they were not at the polar. Oops. After all that work. Oh, sorry. Okay, we're running out of time, so let me just go on to this next one. So there's Jesse developed another assay for the integrity of the membrane. Our model specifically predicts that the membrane will depolarize before the first deformation of the cell morphology occurs. So he's making use of the fact that the fluorescence molecule thioflavin T, which is normally used in unfolding protein assays because it binds to hydrophobic surfaces and fluoresces. If you treat whole cells with that, you don't see fluorescence until the membrane is compromised. And it turns out that hopefully this particular movie has crashed numbers of times. Okay, it doesn't. Okay, so thioflavin T is present. This is very near the end, within seconds of triggering. And what you see is, so it's fairly uniform in the cell long before the sudden explosion at one pole. We have many, many movies like this. Okay, so what time am I at now? Okay, good. So I've got to briefly take a divergence because it turns out after we started this crusade to understand the lambda holin, which I view as the most important protein in the most important organism in the world, it turns out that if we had listened to lambda was saying, but wait, it turns out there's two functional types of holins. So we call the lambda holin canonical because anything in lambda is canonical. But it turns out that there are also pinholins, which make tiny holes. So lambda told us this because its nearest cousin, one of its nearest cousin, I've actually got an untied shoelace. This is what you do when you get to be 78. So the lambda, the phage 21, lambdoid phage, the lysis cassette looks identical to lambda. But when you look at it, it turns out it has a holin that's only got two transmembrane domains. And its endolysin is a normal T4-like lysozyme, but it has what appears to be a transmembrane domain or a signal sequence at the amino terminus. So that was a shock. If I trip and break my neck, then I'll... So, and I'm not going to go through this. This is two seminars is really amazing work to me because we document in two different types of membrane dynamics, membranes jumping in and out of membranes. It's just, transmembrane domains jumping in and out of membranes. But the bottom line is that there is this, this transmembrane domain comes out, this transmembrane comes out, and then what that does is it releases and activates the activity of the enzyme. And there are multiple papers. I actually had a really cute, maybe not so cute, comment from one of the reviewers who may be in this room, said that he really admired our work, but he thought we might have more reviewers than readers. So this is a lot of very high-profile papers about a very small thing, but it was really high cool factor in my opinion. So I'm just going to summarize how this works. I'll flip through the whole thing. So the bottom line is it's the same story, it's just a slightly different musical score. So the holin still accumulates, eventually forms rafts at its critical concentration. At that point, the only hole-forming domain is the C-terminal transmembrane domain, and you end up with, instead of one or two very large holes, you end up with about 1,000 pinholes, heptameric pinholes, and we have cryo-EM tomography, cryo-EM, negative stain EM, and also molecular dynamics modeling, and also all sorts of biochemistry to demonstrate that this is right. So you end up with a heptamer of the S105 protein and about 1,000 of them. Now the problem is that's too small to let the lysozyme out, but that's not a problem here because what happens is when these form these holes, it depolarizes the membrane, and that allows this transmembrane to come out of the membrane, which activates the enzyme. So it's still a raft critical concentration dependent story. It's just a different choreograph. So as shown by this parallel, here is the canonical system we're talking about, and here's the new pinhole system. The only thing that changes is the size of the rafts and the sizes of the holes. Everything else we say is the same. And the genetics of the S21 are equally convincing as they are for lambda. Okay, and we should have noticed this early on because it turns out what you'd expect, since in this case the endolysis is not coming out through one big hole, it's all the way around the cell already. So you're looking at an isotropic destruction of the cell wall, and that's what you see. I'm going to take a risk and try to run this forward. So the boxed ones are going to all go round before they blow up. And notice also that only 7 of the 10 cells that were appearing here did this. And this tells us something we'd already noticed, that the pinhole systems are not quite as well timed as the canonical holin systems are. So they're not. I mean, if you did the lambda S protein with this, they'd all be lysing within a minute or two. So it's not quite as well controlled. So in the last 5 minutes here, we're going to talk about spanins. 2 of the spanin meisters are here. I think is I don't know, is he there? Is he there? Manoj, are you there? I don't see him. Oh well, he's in Shankar's lab anyway, so maybe he felt like he knew it all. So this is the 4 people who did this work. So Liz Summer did the bioinformatics, and everybody else did the genetics and biochemistry. This is the weirdest gene in the world with the weirdest proteins in the world. So the bottom line is the major protein, the RZ protein, has a transmembrane domain and about 100 amino acids of almost totally alpha helical protein. And then embedded in this different reading frame is the RZ1 protein, which has only got 40 residues in the mature form, populated by prolines, and has an outer membrane lipoprotein signal, which means, for those of you who are not familiar with lipoproteins, oh, by the way, before I get to it, so it turns out that phage T1 has the same system, pinholin, SAR endolysin, but it has a different type of spanin. In this case, this was Rohit's baby while he was in the laboratory. It does have a lipoprotein signal, but it's all beta sheet prediction and has a C-terminal transmembrane domain. So let me just show you what that looks like in cartoon form. So the bottom line is, in one case, you have a protein complex that spans the entire periplasm. In this case, it's 2 proteins, the RZ1 protein and the RZ protein. They happen to be covalently linked by disulfide bonds. In the case of T1, you have a single molecule with a completely different topology, and it's all beta sheet. So notice that in both cases, they're threaded through the peptidoglycan meshwork and therefore cannot diffuse laterally. And that, we think, is how they're controlled. So the bottom line is, well, first of all, we had to show that they're required. And so I will, here's to speeding things up. So I was, all of these are without the spanin. Come on. Can I make it go faster? There we go. Eventually, they all turn into spheres. So if you don't have the spanin, but you have the holin endolysin, you get the hole formed, the peptidoglycan is destroyed, but the outer membrane is left intact, so it turns into a sphere. And we actually have a picture of that. This is the same thing for pinhole, the same story. So this is a cryo picture of one of those cells that has no spanin. The outer membrane is intact. And here, the nice thing is you can see, here's the inner membrane, and there's the actual S-hole. And so we caught it in the act. One of the things you learn from this is that absent physical stresses, the outer membrane is capable, if left alone, is capable of withstanding the entire osmotic pressure of the cell even without the peptidoglycan. Okay, so this is we think what happens either with the 2-component spanin or the single spanin, you accumulate, but you're frozen as long as the peptidoglycan is intact. Then once the endolysin degrades the peptidoglycan, quaternary rearrangement, probably oligomerization occurs, and then that leads to collapse of the membrane. And these 2 models, we don't have any evidence for these for the actual formation of these models, but we do have evidence for the collapsing condensation of these domains. And if this looks like viral membrane fusion proteins to you, if you're into that kind of thing, they look like the same to us. So one strong position of prediction, if you extrapolate this into 3 dimensions, what this says is that during lysis, that's the last step mediated by spanins, you ought to segregate the periplasmic contents in vesicular form, and therefore at least initially, the periplasmic contents do not participate, they're not released. All right? So that's one way we can test this model. But first, this is to show it, fusion occurs. So this is Manoj, and who else did this with you? Oh, he's not here. Not here, right? Okay. So the bottom line is you can get we found a trick for putting the spanin periplasmic domains on the outside of membranes. We don't want to go what that trick means, but it allows them, by putting different colors in different vesicles, we can now see directly whether or not those domains are capable of fusing the membrane, and they are. And if you have either defective mutants or no spanins, you get stable absorption, but no fusion. Okay, so our lysis gene roster, two types of holins, two types of endolysins, and remember the canonical endolysins, there are 4 known types of those, and there's 2 types of spanins. I don't know why, there's all these different possibilities, and we've either found in nature or found or constructed in a laboratory almost every combination of these three components. But that's not all, and we promised to just mention antiholins. So about half the holin systems that we've studied have antiholins, and I don't have time to talk about what they do, but the bottom line is in each case, one antiholin inactivates one holin for the purposes of the lysis clock. And the most interesting one is T4, which has, as you remember, it actually has a cytoplasmic domain and a periplasmic domain. It turns out there are 2 different holins, or antiholins, a soluble one and a periplasmic one, and they bind to these different domains. And even more remarkably, that complex between the antiholin and the holin in the T4 case, that complex is sensitive to DNA in the periplasm. And so the model is that if you have a secondary phage infecting its DNA, the DNA goes in, and if you prevent that DNA from getting into the cytoplasm, it then can act as a harbinger that a particular T4 infected cell is in an environment where it's not a good idea to lyse because I'm being super infected. So DNA binds to that complex, stabilizes it, and prevents the holin from aggregating into holes and then triggering lysis. So it's an interesting story on its own right, and very good crystallography by Jim Saccatini's lab. Anna Krieger, in particular, backs that up. Okay, so this I want to show you just preliminary experiments, trying to show whether or not it's true that after spanin function, the periplasmic contents are segregated. So in this experiment, we secreted the superfolder GFP to the periplasm using a signal sequence, and then we just let the lambda take its course. This is a regular lambda lysis with all the genes. So we get to watch, now we're watching GFP in the periplasm, so it should stay uniformly distributed in the periplasm. There's not attached to any lysis protein. So it runs slower in this system. So what's happening is, we think very near the end of lysis is that as the petroglycan is destroyed, the periplasm is squished out into the holes of the cell. I don't know the physics of it, but as you can see, at least in both cases, let me back this up. One of them kind of fell apart after a while, but all of the intensity that was there in the periphery is now there in those 2 vesicles. And just to make sure that this isn't an artifact, we went on to show that if you get rid of the spanins, so now there's never going to be a fusion of the inner and outer membrane. You just have holin and endolysin function. Same experimental detail. So now, of course, you end up with the spherical cell, and again, without the peptidoglycan, for some reason the periplasm collapses and is all squished into one domain. The one part we're not quite understanding is why that domain is leaking SGFP back into the cytoplasm. It might be that there are some small S holes, maybe most of the holes of the holin is in large holes and maybe a few in small holes, so it allows GFP to leak back in, but we haven't done enough experiments to to deal with that yet. Okay, so the last slide is what I call lysis epistemology. Why do holins exist at all, besides to give me 40 years of funding? That seems good enough to me, but so the simplest idea is that all you need to do lysis is put a signal sequence on your endolysin and secrete it, and it'll do whatever you need to do. And so that's possible. And it turns out that in one of the most colossal hypothesis canceling that I've ever been, I looked at the sequence of the mu genome. Mu is one of the classic phages, one of the last ones to have its lysis studied, and there was a clear holin and anti-holin right next to the lysis genes. So I actually labeled in a review, this is likely like mu, holin, and anti-holin. Turns out they're not related to lysis, and in fact, mu has an endolysin and it has a spannin system, but it doesn't have a holin. And in fact, in this case, the mu system uses an anti-endolysin that prevents the activity of the endolysin after secretion. So this is work that's being done by Jolene Ramsey, who just set her lab up at Texas A&M and is carrying on a lot of my multi-gene lysis work. And even more simple is the idea of why not use a single gene lysis system, autolysis, and so what is that? So our lab works on both types. So we've been talking about the endolysin holin system, which we call multi-gene lysis. But in all small phages, like small single-strand DNA phages and small RNA, single-strand RNA phages, they accomplish lysis with a single gene. Sometimes these proteins are genes that inhibit known steps in the cell wall biosynthesis, and sometimes not. But whatever they do, there's no enzyme carried by the bacteriophage, and so instead these are autolysis events. And in work that we did many years ago just for fun, we showed that if you knock out the lambda holin and endolysin and replaced it with the E gene of Phage-174, which we know works by inhibiting peptidoglycan biosynthesis, lambda makes perfectly good plaques. So at least at the level of plaque formation, the lambda E gene complements the holin endolysin defect. So, but yet, so far as we can tell, all double-strand DNA phages use holin systems, and I suspect there must be powerful evolutionary forces to make that happen. Again, we're back to the picture of the people who actually did it all, and I appreciate your patience.
Anyone's here is free to use the mics for some questions as we're waiting online.
Thank you very much. It's fascinating. My mind went on when they are bursting. Is there any energy dispense, and or can you put these under hypoxic or hyperoxic condition to see whether they change the timing of the bursting?
The only thing that matters is the membrane potential. But there's a lot of ways you can mess up the membrane potential. For example, even in Shankar's days in the laboratory back in the 1800s, we knew for a long time that if you infect E. coli with anything, any lambda, T4, any phage, you can't stop the shaker flask. You can't spin them down. And that's because if you stop the shaker flask in a dense culture like OD.2, the oxygen tension drops immediately because it's being sucked up by all the cells. So the bottom line is anything that affects the membrane potential, and we've shown using that spinning bug experiment that we show, you can actually use that to measure the membrane potential at which time the holin triggers. It turns out it only requires a 30% reduction in membrane potential from about 150 millivolts down to like 100, and that's enough to trigger. So you can do these experiments in anoxic conditions as long as you're providing the membrane potential by fermentative methods. So it doesn't matter how you, as far as we can tell, the only thing that matters is the membrane potential.
Because they behave somewhat like mitochondrial oxidative process.
I agree. You know, what's the name of that? BCL was one of the proteins that are in the mitogenic pathway. It turns out the BCL protein complements lambda S for hole formation. And so my name is on a paper that says those mitochondrial proteins are holins. I don't know why I allowed that to. It's not true because they don't have the key element of the holins is they have to be triggerable by cyanide or by energy poison, because we think that's integral to the mechanism. And the whole timing thing, nothing will work without that. But I couldn't turn down getting a paper in IMBO.
Very nice, thank you.
I have a question over here.
Yeah.
Hi. How do the holins go to the curvature or to the poles?
They don't always do that. I was sort of pissed off that 80% of them happen at the poles, but they don't always do that. I think there's a decision early on, because when they show up elsewhere, they don't show up at the poles on either one. So I suspect there's some sort of sorting event that occurs. It's not lysis specific. There's still a lot of controversy about how any protein finds the poles.
Thank you.
The answer is I don't know.
Well, there are no questions online, surprisingly. Lots of people watching. So, Dr. Lorsch, do you have, well, I'm sorry, wait, something just came in? No, that was nothing. That was my test to see if it was actually working.
Actually, since this is a lecture, there is actually a test. And so,
Yeah.
Let's thank Rai for a fantastic tour de force talk. And there will be a short reception outside for those of you who are here in real life. And those of you who are virtual, you're on your own. You can go get a cookie downstairs or whatever you want to do. But thanks again, Rai.